that going. Alrighty, so good morning and welcome to the very last of our live lab walkthrough sub-series this fall at Fourth Space. We're joined today by Yves Gélinas, professor in Chemistry and Biochemistry and Concordia University Research Chair in Environmental Chemistry. Today we find ourselves at Loyola, very exciting, at the LEGO laboratory, part of the Geotop Strategic Research Network and the Chemistry and Biochemistry Department at Concordia University. Yves Gélinas, who joins us, teaches in Analytical Environmental Chemistry with particular interest in studying the health of coastal aquatic ecosystems such as the St. Lawrence Estuary and Gulf in relation to climate change and anthropogenic activities. So on that happy note, I'll pass it over to you, Yves. Good morning, welcome. Oh, we can't hear, no sound yet. Doug, Mackay, we've got no sound.
I have to mime.
Okay. Yeah, we got it now.
All right. Very good. Thanks. Should I go? Yeah.
Take two.
So one of the difficulties that we have here is that we have three different rooms. So I'll take you to the first room as a start. So this is the lab where we actually store samples and do a little bit of preparation. And so we go at the end. Thank you. Light should come up. And so as Anna said, I work in environmental chemistry. Well, actually, I've been hired as an analytical chemist, but don't tell anyone because I like to say that I'm doing environmental chemistry more than anything. And so what we do really is biogeochemistry. So we're actually looking at the interactions between the chemistry, the biology, and the geochemistry. So we're a little special here in terms of specializations, area of specializations. We work in the environment. We're studying the chemistry of the environment. And our playground is the St. Lawrence Estuary and Gulf. So if you can zoom on this map, you have Montreal here, Quebec City is right here. And this is the St. Lawrence. So this is a river here that goes all the way to Quebec City. And when you reach Quebec City, the water becomes saltier and saltier. And so the estuary starts at Quebec City and goes all the way to this area here at Saint-Anne-des-Monts. We're also looking at the Saguenay Fjord that's right here, and at the whole Gulf of St. Lawrence, which is the largest enclosed or semi-enclosed sea in the world. And so we're looking at this system using two different angles, one that is more applied and one that's more fundamental. So on the applied side of things, we're looking at contamination because of human activities. And so, of course, the St. Lawrence Valley here, there's a population density is very high. So human activities produce a lot of waste. and so contaminants as well. So one of the things that we do is looking at contamination by petroleum products. Right now, there's no overall contamination in the St. Lawrence, well, no, very little overall contamination by petroleum products in the Estuary and Gulf. But if you remember a few years ago, 5-6 years ago, the big accident that happened in Lac Mégantic, where there was a train that actually exploded in the city. The train was transporting petroleum products from the prairies. Now there are plans to transport more and more petroleum products through the St. Lawrence Seaway and through the estuary all the way to whichever country it's going to. And so what we're doing really is looking at hydrocarbons that are naturally present in the system, because there are hydrocarbons that are present in the system naturally, and also looking at potential contamination, developing method in order to be able to detect this contamination, the extent, for how long it's staying there. So that's one of the things we do with organic contaminants. But we're also working a lot on the more fundamental side of things, looking at the cycle of organic carbon in that system in relation with CO2. And so one of the things that people don't know about the St. Lawrence Estuary is that close to Rimouski, just in this area here, there's a zone in the bottom waters that is labeled hypoxic, so a zone where there's very little oxygen in the bottom waters. And we're looking at the exchanges between CO2 in the atmosphere and the sediments. So we do a lot of work with sediments. We do a lot of work on the water column as well, looking at carbon, organic carbon in the water column as dissolved species, as particulate species, just to try to understand how carbon moves from a reservoir to the other one. And also because sediments are a sink for CO2, the only long-term sink for CO2 on this planet. So CO2 from the atmosphere is fixed in the surface water as phytoplankton. So they're plants. Phytoplanktons are plants that are growing using the energy of the sun, fixing that CO2 as organic matter that degrades, that, you know, these organisms die. Eventually, this organic matter sinks to the bottom, to the sediments, and it gets buried in the sediment. And so this is the only natural way that the planet is actually using to remove some of the CO2 from the atmosphere. So we're looking at this transfer mechanism, which in this system is highly impacted by, again, human activities, either because of the discharge of organic matter directly from our agricultural activities, or because of the nutrients that also come from agricultural activities and wastewater that are discharged by the river, that fertilize this area here. And because of the fertilization, there's a lot more organic matter being produced and a lot more organic matter sinking to the bottom, causing hypoxia and leading eventually to more carbon being buried in the sediments. And so we're interested in these different things, trying to understand the cycling, the controls on the cycling, and more importantly, we're interested in trying to understand what is going to happen with climate change when conditions change. There could be eventually, we call them catastrophic, but catastrophic situations where we could reach a point where there's no turning back, and the system, the health of that ecosystem may degrade quite a bit. So that's our playground. What we do, I've set up a few stations where I want to show you, because it was impossible to go on a ship. The ship is based in Rimouski, which is right here, actually. And so whenever we go out in the field, when we collect samples, we have to take our equipment, go to Rimouski, then we get on the ship. The ship is the Coriolis. You see on this picture here, you see part of this ship. And then we have our equipment to try and collect samples, the sample that we need for our analyses. So this first station here is water sampling. And so we're collecting sample from the water column with what we call a hosette. This is simply a rack that contains 12 bottles that goes down at different depths, and at the depth that we want to collect water from, the water just closes and we can then bring it back to the surface, to the ship. And so once it's on the ship, we simply transfer the water into a bigger jug and we measure it and we filter it. This is a filtration apparatus. And so we simply put a filter right here in between. We filter the water because we separate the dissolved species from the particulate species. So for the dissolved species, we simply have a small bottle like this in which we collect the water. And from this water, we also collect separately particulates. So this is the filter. These are examples of the filters that we use to collect the particles. And as you see, there are differences depending on where we are in the St. Lawrence. Sometimes there's more particles, sometimes less. And the reason why we do this is the reactivity of dissolved species is very different from the reactivity of the particulate species. So it's important for us to know, in terms of organic carbon, to know, what these particles contain in terms of particulate carbon, where they come from, what is their reactivity, and so on and so forth. And so we then run analyses on these samples. Just to give you an example of how difficult it is to do these types of analyses, I've just set up, this is an equipment that we have here that is used to measure the concentration of dissolved organic carbon in seawater. And just to give you an example of how difficult it is, I have set up here. So this is 1 liter of water coming from the St. Lawrence estuary. So if you remove the water, if you dry it down and you just look at the salt, this is the amount of salt on the left that 1 liter of bottle contains. So this is the salt. And when you look at organic carbon, I don't know if you can see there's 1 grain here. And I've weighed these accurately. That's how much carbon there is dissolved in this bottle as opposed to the salts on the left. And so we're interested in this carbon, not only in terms of total carbon, but in terms of what type of molecules it contains, what is their reactivity, and so on and so forth. So we do analyses at a very, very low level. In fact, it's very difficult to show you what we do because most of what we do, we simply cannot see it. Okay, so this is for the water. That's an example here of the number of samples that we collect during one cruise. That was the cruise we went out again in August of this year. And so we collected all of these water samples. We analyzed them for molecular composition, for stable isotope composition, for quantity as well, to be able to do mass balances, to be able to know where the organic matter is coming from, what its reactivity is. And that's it. So a lot of work to do in the lab. That's for the water column. That's only one part of what we do. We also, as I said, since we're interested in the transfer of organic carbon to the sediments, we also collect sediments when we go there, and we have to move to the next station. So I'll pass right in front of you, sorry.
I have a question while we're speaking, which is, what is, especially around kind of measuring the environmental changes that are happening in the facility, important work to be doing as there's all of this resistance to the new pipelines. What is the scale of time that you're working with when you're trying to evaluate those changes? Like, do you have a control that you're measuring against, or?
So what we're trying to see how the system has been evolving in the past years. So for instance, one of the main concerns in the St. Lawrence estuary is the hypoxic zone, because the concentration of oxygen is so low now that it directly affects life. Fishes, there's only one type of, I don't know what the name in English is sebast, only one type of fish that can still survive in these environments. And so since the beginning of the century, measurements have been taken, and so we're monitoring the decrease in oxygen in that system, and we can pretty much, well, I mean, we're fairly capable of predicting how long it will take before the system becomes completely anoxic, which will lead to irreversible changes. It's difficult to project ourselves like tens and tens and tens of years ahead, but we can predict what might happen if the situation doesn't change, if the conditions remain the same in the past over a time scale of maybe 10 years, 15 years, 20 years. More than that, it all depends what is going to happen, both on the parameters that we control, contamination coming from the St. Lawrence Valley. If political action is taken and there's less nutrients getting into that system, there will be drastic changes probably, but also another reason for the presence of hypoxic conditions in the bottom waters is changes in the global oceanic circulation. So currents change, and that is led by changes in climate, the warming. And so because the currents are changing, there's not much we can do here on the local level. Well, there is something we can do on the local level, but it's not going to affect how currents are changing. And that is happening on a longer time scale, which is harder to predict. So that's what we do, more on a local, not local, but short time scales. But the one thing I have to say is the data we're collecting is being used by people developing global models on, climate change and so on and so forth. I'm thinking Dave Matthews, for instance, in the geography department is using these models to try and predict on a longer time scale what might happen. So this is the sediment station that I prepared shortly just to show you. The way we collect sediment when we're on a ship is using what we call, there are different ways of doing it, but what we do most of the time is using a box core. And so this is a picture here of the box core before it goes down. And so it's tied to a wire. It goes down all the way to the bottom. Once it reaches the sediment and it's into the sediments, there's a closure here that closes down. And when it comes back up with the sediment, it's usually full of sediment, so about 40 centimeter high of sediment in the box. And then we take cores such as this one here that we just simply put into that box core to subsample a fraction of that sediment. And I just, the last time we went, well, that's 2 years ago when we went, we collected a core that we left right here in its tube, in its core, just to give you an idea of what it looks like. This one has changed since we collected it. It became completely dark. That's the normal color at the surface of the sediment.
So the whole thing was gray when you first collected it?
Not all of it, but usually it's gray and then it becomes black at the bottom. Black means anoxic.
Oh, okay.
So because there's still oxygen at the surface, grayish is more oxic, and then it becomes anoxic. Usually there's a gradual changes to hypoxic conditions. But what I want to show you about this sediment, the nice thing about looking at organic matter in sediments is that we can use the organic matter to reconstruct the history of what happened in that system, the changes that happened in that system, because you realize that the sediments accumulate with time, right? So particles just settle to the bottom and they just accumulate. They're mixed a little bit, but not that much. So if you run analyses of any kind, if I'm looking at a biomarker that shows, for instance, productivity in the surface of the estuary, and I look down, I subsample different layers of that sediment, I'm actually going back in time. The deeper I am in the sediment, the longer back in time that I'm looking at. So
what would be an example of a biomarker that you would look for?
So we look at lipids, mostly lipid biomarkers. They're molecules that we all synthesize. Phytoplankton cells also synthesize these lipids. Fatty acids, for instance, some are specific to certain type of phytoplankton. We're interested a lot in the degradation of organic matter. So we're looking at bacterial biomarkers. So bacteria are very special in that they synthesize a very specific fatty acid that only bacteria synthesize, but all bacteria synthesize it. So most of the bacteria, I should say, synthesize it. So if you're actually looking at that biomarker, you have an idea of bacterial abundance at least. And we're doing something even more. We'll talk about this maybe a bit more downstairs, but we're looking at the stable isotope, carbon stable isotope signature of that specific biomarker. So we're adding another level of information to that analysis just to get a better idea of exactly what did that bacteria degrade when it was alive. And so we have a better idea then of what type of organic matter was there when the bacteria was alive and synthesized those fatty acids.
And so if this kind of represents geologically like there's a range of time in this tube, how narrow do you go as a lens into this? Like do you?
So we did, I'm showing you this as well. This is how we collect it. So we designed this, we built it, actually, it's a student, not me, it's a student who built it to subsample the sediment. So what we do is we simply imagine there's sediment in there. We just go down like this, we extrude the core using these little spacers. And so this is 0.5 centimeter. So at the surface, we collect depths of 0.5 centimeter because there's more reactivity, there's more things going on here. So we simply go down like this until the top of the sediment reaches the surface here. And then we use tools such as this one to just simply subsample the right layer that we transfer to a tube. This is the example of a dry sediment that we collected exactly in that location. So once it's dry, it looks like this. Behind you, there's a freezer full That's one of our 4 freezers filled with mud that we've collected. We have mud from all over the world, Antarctica, Arctic, the Arctic, and so on and so forth. And we use my children's little spoons. You know, I don't want to lose these spoons. We've been using that spoon for these 2 spoons for as long as I've been going out there. They're not using them, obviously. So that's how we collect sediments. And then we run different types of analyses, both for contaminants, for also interaction between minerals and the organic matter, just trying again to understand the cycling of organic matter in that system. So that's it for this room. We're going to move to the other room across after you. Thank you. So welcome to the LEGO lab. I'll let you go first. Yes. Thank you. So this is a chemistry lab. This is where we do most of our sample preparation. It's pretty messy, but you have to understand that we're working with mud all the time. And so I cleaned it a little bit, but not that much, because I want to show you what the real chemistry lab looks like. It's a little on the messy side of things, but that's really, you know, when students are working here, that's what it looks like. And so obviously, because we're dealing with organic compounds, we're using organic solvents to extract the lipids. For instance, when I was talking about lipidic biomarkers, we do all of this in a flow hood. This is just a station where we actually do the separation. So once we extract whatever we're interested in, the molecules we're interested in, we have to separate them, to clean them, to get fractions that can be analyzed directly on the instruments that we have downstairs. This is one of the stations where we do this type of thing. Behind you, we have, again, freezers and fridges full of samples. Well, if you want to see what so that's just part of the samples that we have that are currently being analyzed, actually, currently being prepared and are ready to get analyzed downstairs. Well, not all of them, most of them.
This processing that you're describing, like,
yeah,
from start to this stage, what amount of time gets invested in the lab on a single kind of vial of water?
That's the most time-consuming step, actually, because once it's ready, all you have to do is analyze it. So you just inject it into the instrument, you get your not response, but result right away. This takes time. It depends what you do with them. But we prepare the samples in batches, but for instance, just to prepare one batch, it can take as much as 2 to 3 weeks before you get a sample that's ready to be analyzed. It's not the same thing on the other side, what I've shown you on the other side with the dissolved versus particulate. That's quick. This here, and that's done on the boat. Here, this is done in the lab, in the flow hood, and it can be time consuming depending on what you're doing. Because usually we're just going to close it if you don't mind. Yeah. Because you have organic solvents in there, you don't want to leave it open too long. Yes, it depends what you want to do. And usually when we extract the sample, going out at sea is very, very expensive. Obviously, it's $16,000 a day when you go on a boat like this one, just to rent the boat. So we have to write grants. We have to, you know, be successful in writing a grant and getting the money. So we try to optimize as much as we can, both our time on the boat and our samples once we come back. So every time we do an extraction to look at one particular thing, we always try to make the most out of the sample by looking at everything that we're interested in. And so when we do something like this, it's a multiple step separation, preparation, cleanup, and so on, and that takes time. Yes. So the last thing I want to show you here, the rest is not really, so we have instruments to do, it's called gas chromatography measurements. And those are pretty basic instruments that we use just simply to make sure that the samples are well prepared. Our analyses are done mostly downstairs. The only thing I want to show you maybe is this box here. If Alexandre, my student he's going to be defending his thesis next week, no, in 2 weeks from now, he spent 3 years, his hands in this box. This is called the glove box. Yes, I know, it's torture. He decided to work on this project. I usually, suggest a few projects to students and they decide what they want to work on. And he decided to work on looking at the anoxic interactions between organic matter and minerals in the anoxic zone where there's no oxygen. And so anytime you try to reproduce what's going on in the natural environment, we're trying to mimic the conditions that exist. And so if there's no oxygen in the sediment, he has to do his preparation without the presence of oxygen. And so this is just a glove box. It was very funny because his partner is in arts and she had silk gloves, black silk gloves, and to make sure that he would slide his hands without, sticking and sweating and so on. He was wearing those very tall silk gloves with a feather at the end. So it's very cute. Anyway, so this was where Alexandre worked for almost 2.5 years, and successfully, because he's defending his thesis in 2 weeks from now. We're done here, unless, the rest is just regular equipment that we use on a regular basis in the lab. And so now we have to move downstairs. I don't know how we're doing with time. We're good? Have to move downstairs where we have the equipment that we use to actually do the measurements for which, following the preparation of these samples, that gives us the answer we're interested in. Thank you.
Anna, you can spotlight the card. We're going to go down some stairs.
Everything is good? Yeah. That's good. So the reason we're going downstairs is because we use instruments that are called mass spectrometers to do our measurements. And these instruments need a special temperature, controlled power, and so on. And so we have in the department a center that's called CBAMS, the Center for Biological Applications of Mass Spec. I'll hold it. Is that okay if I hold it? Is the sound good? I'll just clip it. You'll clip it in? Sorry. Good. Sorry. And so we have that mass center, and that's where all of our instruments are installed. So it is a little bit, sorry for this, but that's because these instruments are under vacuum and we need pumps to actually pump them down. I was going to stop just behind you just to show you a first. It's not really interesting. It's like looking at a car. It's boring. But this is the first instrument here that we use to measure metal ions, either soluble or dissolved or in the sediment. And so because we're interested in the interactions between the inorganic world and the organic world, we have to know the composition of the inorganic materials, the minerals, and we do this by looking, among other things, looking at the elemental composition. So this is called an ICP-MS, so inductively coupled plasma mass spectrometer. So they're all mass specs. It works with a plasma. So plasma is simply a torch heating up at 7,000 degrees just to decompose the sample and to ionize the different metal ions, dehydrate them and ionize them, and then they're being analyzed and detected by the mass spectrometer and the detector. So that's the first instruments. We don't use it that often. This one belongs to CBAMS. It's not my instrument. These were instruments that we purchased for our own use. This is a type of instrument that even chemists are not very familiar with because it's very specific to the world of geosciences. So it's called an IRMS, so an isotope ratio mass spectrometer. And so just to give you a little bit of background to understand, if you remember your chemistry courses from either high school or CJEP, with good memories, I'm sure, you probably remember that carbon has a mass of 12, and you might remember that there's also another isotope of carbon that has a mass of 13. So 1% of the carbon atoms have a mass of 13 rather than the mass of 12. And I'm not talking about radiocarbon here, radiocarbon that is used to date in anything that has carbon in it. That's carbon 14. That's a radioactive element. I'm talking about stable isotopes. That 1% is actually 1.1 with decimal points, 4 or 5 decimal points. Is the average ratio 1%, the average percentage that is usually given to anyone who's interested in these things. But with this type of instrument, we're actually able to detect changes in that ratio to the 4th and 5th decimal point. And these changes are due to different processes that happen in the environment. So anytime you have a reaction going on, let's just say, a good analogy is if you're moving a friend from a house to another one, and you're going into a room and there's tons of boxes there, you have big boxes and small boxes, your first strategy will be to pick up the small box, right? Because it requires less energy. It's the same thing for chemical enzymatic reaction going on. If you have the choice between a mass of 13, carbon with a mass of 13 versus carbon with a mass of 12, the enzymes and the reaction that will go on will select mass 12 over mass 13. So you get what we call fractionation. And this fractionation, this change in the ratio between carbon 12 and carbon 13 varies depending on different type of processes, the source of the organic matter, all sorts of things that we play with a lot. Bacteria, for instance, we're looking at this ratio in the specific biomarkers that are specific to bacteria, simply to get an idea of the source of the organic matter, either terrestrial or marine, that the bacteria are degrading in the sediments. And so this is the instrument that we're using. That's the small mass spec. It's not a very sophisticated mass spec because we're looking at small molecules. We have an elemental analyzer to look at the ratio for an entire sample, a bulk sample. And we did it again. Sorry about this. I'll leave. And we also have a GC, so a gas chromatograph, that does the same thing, that looks at the stable isotope ratio, but on specific compounds. So this is where we do the analysis of biomarkers plus their stable isotope composition. And, well, I'm not going to talk about this. These are the types of analyses. I don't think I need to go through the chromatogram. It's not going to be very interesting for you. And so the last instrument that we have is on the other side. So we have another one here that does the same thing. It's just a different company that does the same thing as the one I've just shown you. This here is called also a mass spec. So it's a gas chromatograph mass spectrometer. What it does is a little different. It's not looking at the stable isotope ratios. It only looks at the identity of the molecules. When we extract organic molecules from a sample, we extract like thousands of molecules, a lot of molecules. And so we have to identify these compounds. And so one of the things that we do is run our sample on a mass spec like this one that not only separate the different compounds, each peak here corresponds to one compound. That's done by the GC, the gas chromatograph, that separates the compounds. But each one of them is then analyzed with the mass spectrometer to give us the identity of the compound based on its fragmentation pattern. It breaks down into pieces, and the pieces are very specific to the compound. We have a data bank, and we just search through the data bank, and we're able to identify a lot of, not all, but a lot of the compounds in this way. And so
for example, what we're looking at here, could you just off the top of your head identify things?
yes, well, at the top of my head, usually what I would do is a search, but because I've done that often, I know that this breakdown pattern. So we're actually looking right now, which one are we looking at? So we're looking at this small molecule here. That's why you see the serrated red lines. And so usually when you have a breakdown like this with specific ions, 57, 71, 85, we know that this is a hydrocarbon. Okay. I cheated a little bit because I took a standard. That's a standard, so it's very clean. But even for real samples, we can detect and identify them with the eye. But what we do most of the time is just go to the data bank, and we just get the best match possible, and then it gives you an idea of the quality of the identification. So depending on the percentage you're getting, you know that yes, it's 95% certain that we have this compound, or it's only 30% because there are slight differences and we're not certain. So
when you're in that kind of gray zone, does it mean that it's not usable, or?
it depends. Yeah, I mean, very often, yes, it's not usable, but it gives us the breakdown patterns here, gives us idea on the type of compound it is. So even if we're not certain that it's that specific fatty acid, we know that it is a fatty acid. And so we can still use that result to try to understand a little better what's going on, but maybe not as specifically as if you knew exactly what molecule it is and what biomarker it is, if it is a biomarker, and so on.
So in terms of this, what would be like the gold standard, like A+ sample? Like, is it sort of an accident when you land on one, or like?
Well, usually we have different quality of samples, right? The fresh sample. So for instance, the particles that I collected that I've shown you upstairs, those are fairly fresh because they're particles raining down from anything that happened in the surface. So those are high quality. So normally you're able to identify maybe 25, 30, 40% of the compounds in the sample like this one. But when you go down in the sediment, the longer those molecules have stayed in the sediment, and the higher the potential for chemical alteration, either through biological reaction or through simply like chemical reactions. And so because you have these slight alteration, one functional group that changes or it becomes something else, and on a full molecule, it becomes more and more difficult to analyze and find them. And so most of the time, because we're working with mud, the sample that I've shown you upstairs, the sediment, 40 centimeters, depending on where you are in the estuary, it could be a sample that's about 200-year-old to a few thousand years old. So when the sediment has sat there for that long, then it's difficult.
I know you don't do radio dating in this lab, but is that
we could
rough estimate? You generally have a sense when you take those thin slices and how far back?
So we do, because other people have worked on this. The St. Lawrence has been studied in the past fairly often, and one of the first things that people do is build an age model. We call this an age model that simply tells you, okay, if you're 30 centimeter deep in the sediment, at that point, you know that it's a sample that was that accumulated between, I don't know, 500 years ago and 550 years ago.
And
so
the movement of the river, does it kind of fluctuate in there?
So it does make a difference, but when we're talking about the St. Lawrence Estuary and Gulf, so the river influences mostly the first part of the estuary, and there the current can change so much, and you have the tides as well, that you can't really look down in the sediment like this because it's not an accumulation zone. You need a zone where sediments regularly and evenly accumulate with time, and so that's further seaward from Quebec City, like it's actually past the Saguenay Fjord. So the estuary is about 20 to about 100 meters between Quebec City and the Saguenay Fjord. And then once you're past the Saguenay Fjord, it goes down to about 400 meters, all the way to 500 to 600 meters close to the Atlantic Ocean. So that's where sediment accumulate nicely and form these layers that we can study. Yeah. So this is an overview of what we do in the lab. Is the timing good? Yeah. So I'll be happy to answer any questions, more questions that you may have.
Yeah, I have some more kind of like big frame questions. I mean, mostly you're working with this tremendous shift of scale, right? It's like you go out into the biggest inland sea that exists in the planet. You collect, and then I think about that shift down to this minute spectral scale, and I think the visual aid you gave us of the liter of water with a single grain of carbon coming out of it. So you're working on one hand with something that's huge and one thing that's really tiny. And how do you translate that in terms of working with partners or collaborators or researchers who are outside of your field? Or how do you see that shift of scale playing a role in how you communicate?
Well, there's a golden rule in our field, is that the process you're looking at must have the same time scale as the time scale over which you're studying that process. And so, of course, there are things we cannot do with certain people who are looking at very, very minute, very fast occurrences or things happening. And so we have to match what we do and make sure that we're looking at the same time scale, because it's impossible to just infer and find relationships between, I don't know, if I'm talking about someone in biology, for instance, versus someone in geochemistry. In our field, we're looking at longer time scales than people working in biology. Just to give you an example, I have, and I'm sorry for the words I'll be using, but I have a colleague who's in biology but doing geochemistry. She keeps saying when you're looking at carbon in the ocean that whales are a piece of shit that no one cares about, because we don't care about the fishes, we don't care about the whales, because in terms of quantity, overall quantity of organic matter, it's very, very small compared to the plankton, compared to the bacteria, and so on. And so we have to adjust, right? I mean, as a geochemist, the whales don't exist and fishes don't exist, even though I know the effect that changes in conditions may have on these species.
Well, and it sounds like, earlier when you mentioned that there's just one species left that can survive in that hypoxic zone, I mean, that they do become in some way a metric of these larger processes that you're studying.
Somewhat, yes.
Yeah.
I mean, again, yes, I mean, that's a good point. But don't forget that we work as teams. When we go out, again, I told you about the cost of renting that ship. We are 13 scientists on that ship. And so usually we try to form teams that are complementary in what they're looking at and what they're studying, so that we can actually find a little, learn a little more about the system, looking at different angles. So the chemical only, we are biased, but we're looking at the chemistry only. We're not looking so much at the biology, yet the biology has a great influence on the micro scale, has a great influence on the processes going on. If the system becomes anoxic, it's because of the activity of microorganisms that degrade organic matter consuming oxygen. So we do have to take into account these things, but our time scale is very different. But even for what we do, we have shorter time scales and longer time scales. We have sediments upstairs that go back to the last glaciation 11,000 years ago. And so we can reconstruct what happened between the last glaciation and now. It would be close to impossible to know what happened on the biological time side of things over that time scale. And so we just have to adapt and be the best. The only thing that we can do is be honest with the interpretation of our data, not make them say more, over interpreting the data, being honest and saying, this is what we find, this is what we think. The rest is speculative. We know it's speculative, but we think that's what's happening.
Maybe I'll just jump in. I see that Joya's popped a question in the chat.
Maybe a couple questions. Would you be able to step just outside?
Sure.
It's a bit louder.
Yes.
Yeah.
Yeah.
Ooh.
Thank you.
Looks like they're going somewhere else, so we'll.
We're just getting out of the sound for a second. I'll let you, but we can take any questions here for you.
Hi, Yves.
Sorry, go ahead.
All right. I see that Joya's popped something in the chat. So Joya, I don't know. Do you want me to read it for you, or you can go ahead?
Yes, okay, perfect. So my question was, because you mentioned earlier how much the days on the boat are planned intricately. So I'm wondering, are there moments where things go, let's say, out of the plan and there are surprises or unplanned discoveries? So that's what I'm wondering. It's a curious question.
No, it's a highly relevant question. Is that okay? Yes. I mean, it does happen very often that things are not happening the way we want. The rosette to sample water, for instance, that we use to collect a water sample, at some point the cable, it's a steel cable, but it got entangled into the, how do you call this?
Propeller?
The propeller, thank you. And it got cut. So we lost the rosette completely. It went down completely to the bottom. It was completely lost. They tried to recover it, but couldn't recover it. And so they had to go back back to Rimouski to get another one. There's a rule also at sea. If there's someone who's in trouble, any ship that's in trouble, then there's a search. And every ship in the area has to then participate into that search. And that happened to us once. It was a sailboat that was missing. It was supposed to be in a certain location at a certain time. It did not report. And so we had to go out and try to see if we could find the ship somewhere, could locate the ship, or evidence that something had happened to the ship. Luckily, nothing had happened to the ship, but yes, it does happen. We have to adapt. That's rule number one, is we have to adapt and try to make the most out of the time that we have on the boat. And we have very, very, very long days. I have a student who is seasick. She knew she was seasick. She was a PhD student, poor Anik. But one time the sea was so rough when we left port that she got sick, and so sick that at some point, I'll have a hard time saying these words in English. The esophagus. So she threw up so much that she started bleeding from her stomach and her esophagus. I don't know how you say this in English. I'm sorry. And sorry? No, the tube that goes from the mouth. Okay, esophagus. That's how you pronounce it. Sorry. So we had to stop everything we were doing and go to Quebec City just to drop her off, and she had to go to a hospital. And so yes, these things happen all the time. She never came back on the ship after poor her. Thank you for your question.
Thank you to you.
All right, unless anybody wants to jump in with something else, I know that Yves is exceedingly busy, and we really appreciate, Yves, the fact that you met up with us early morning today to show us around these 3 labs. Very comprehensive tour and visit. We really appreciate your time and energy on this spent with us. And perhaps since this is the last of our live lab walkthroughs, I will just thank the entire team behind the scenes who's been working on these productions every single week, multiple times a week for 10 sessions. Cecilia and Joya specifically, who've been helping us out, Makai and Doug, we're passing the camera around, and all the students who have participated, either watching it live with us, adding questions to the mix, or watching after the fact on YouTube. We really appreciate all of you. So thanks, team. Way to deliver. That was really fun. And Yves, once again, thank you so much for your time and energy. It was a pleasure to meet you in this way and to take a look around in your world.
Thank you, Anna. It was very fun to participate. And all I can say is you have a team that's very, very enjoyable to work with. So thank you, and it was a pleasure to be here this morning.
All right, thanks everybody. On that note, once again, bye-bye. See you next time. Cheers.